Welcome everyone to Module 9-2, the Audit-Proof Lab. Today we are moving beyond basic laboratory skills and into the world of strategic compliance and risk management. We are here to master the global language of trust, the bioanalytical method validation standards set by the FDA and EMA. By the end of this session, you won't just be producing data. You will be building an unshakable scientific legacy. Imagine you are standing at the base of the world's most impressive skyscraper. But as you look at the waterline, you realize it was built on a foundation of loose, shifting sand. In our world of high-stakes analytical chemistry, that skyscraper represents your research. You might have the most expensive Orbitrap or FTICR hardware, but without a rigorous validation foundation, your data has no legal standing, no scientific weight, and is a tragedy waiting to happen during an audit. I want you to stop thinking of validation as a box-ticking chore. It is the ultimate form of risk management and strategic assurance for your intellectual property. Our goal is threefold: ensuring legal standing so your data carries weight in boardrooms and courtrooms, and achieving regulatory alignment by speaking the universal language of trust used by the FDA and EMA. To build a lab that can survive global scrutiny, we focus on four critical pillars. First, calibration logic, our roadmap translating noise into concentration. Second, accuracy and precision, our bullseye. Third, the stability marathon, ensuring integrity over time. And finally, selectivity and matrix, seeing through biological noise. Underpinning all of this is the 15% rule, the mathematical threshold defining global success. Let's start with calibration logic and the matrix trap. The most recurring fatal error in global audits is preparing curves in neat solvents while your actual samples are complex matrices like human plasma. To satisfy global standards, your calibration must occur in the exact same matrix to account for ion suppression. We are looking for professional linearity, with a goal of an R-square greater than 0.995. Remember, quantification is a measurement, not a high-tech guess. Why do we obsess over matrices? Because biological samples like plasma or urine contain salts and lipids that cause ion suppression or enhancement. If your curve doesn't account for this complexity, your data is a fantasy. We must prove that our quantification is a precise measurement that survives the chaotic variance of real-world samples. Accuracy and precision are the core of our defense, and I use the bullseye analogy to set the record straight. Accuracy is hitting the gold, how close you are to the nominal value. Precision is hitting the same spot every time, the reproducibility of your shots. To pass a global audit, you don't get to choose one. You need both to deliver the truth. Now, let's look at the mathematical law. The FDA and EMA demand that for most quality control levels, your accuracy, mean bias, must be within 15, and your precision, CV, must be under 15%. However, agencies offer a consultant's grace at the sensitivity floor. For the LLOQ, we allow up to 20% error. We also require repeatability tests, ensuring Monday to Friday consistency across intraday and interday runs. Validation isn't just about day one. It's a time stress test. You must prove your analyte survived the journey, including short-term stability on the bench, long-term survival in the -80 Celsius freezer, and the endurance of freeze-thaw cycles. If you cannot prove the molecule didn't fall apart while waiting for the instrument, your numbers mean nothing to an auditor. The biological world is noisy, and selectivity is our shield against that noise. You must provide proof that the peak you are seeing is actually your analyte, and not a metabolite, a degradation product, or a random component of the patient's blood. We must ensure there is zero interference, effectively seeing through the noise of complex samples. In summary, 100% data integrity is the difference between a random number and a fact. Validation is the shield you carry into the boardroom to prove that your numbers represent the absolute truth. By mastering these four pillars, you achieve a level of global compliance that sets you apart as a professional and protects your scientific legacy. We have covered the trenches of calibration and the physics of accuracy. Now, I want to open the floor. Are there any questions regarding the implementation of these standards or how to master the global language of trust? Thank you for your commitment to analytical excellence.